My name is Bryony Pearce and I'm the Research Director for Marine Ecological Surveys. We're quite unique within the field of environmental consultancies because at least a third of our work comes from applied research. And my job is to organise that part of our business. So I'm in charge of getting funding in to do research and coming up with ideas and identifying areas where research is needed. So the commercial side of the business works primarily on environmental impact assessments, and the reason that we need these is because as humans we use the sea for a variety of different activities, including fishing, to supply building materials, for ports and shipping, and all of these activities have an impact on the environment. So what our job is to go in and assess what impacts those are likely to have, and as far as we can to mitigate against them. Before I started my job at Marine Ecological Surveys, I carried out a degree in marine biology at the University of Plymouth. In addition to this, I carried out voluntary work with a project called Proyecto Ambiental, who were carrying out surveys of the marine environment in Tenerife, and I also worked at the Marine Aquarium as a volunteer diver, and all of that work contributed towards me getting the job that I got at Marine Ecological Surveys. I was lead biologist in the Humber REC. Marine Ecological Surveys carried out the survey work for the Humber REC. We analysed the samples in our benthic laboratory, and then we carried out statistical analyses on those sample data in order to map the resources in the area. We also were responsible for the integration of the data, and our GIS department carried out the modelling, the biotope modelling for the Humber REC as well. Benthic macrofauna are animals which live in association with the seafloor. The term macrofauna specifically applies to animals which are between 1 millimetre and 10 centimetres in size. And within benthic macrofauna, there are two components, epifauna and infauna. Infauna are the animals which live within the sediments, and that is predominantly polychaete worms in the marine environment, and they may live in tubes or they may swim between the sediment grains. The other component is epifauna, and those are animals which live on the surface of the seafloor, and they may be attached or they may be mobile. So attached epifauna will include things like sponges and hydroids and bryozoans, and then you'll have mobile epifauna, which will be things like crabs, hermit crabs, and also demersal fish. Benthic macrofauna are studied to look at the impacts of anthropogenic activities on the sea, and this is because they've been shown to be good indicators of stress in the marine environment. It's not that we're not interested in other components of the marine food web, such as cetaceans and seabirds and smaller organisms such as myofauna, but we don't have the resources to study all of these things, and this particular component of the food web is quite easy to sample and quite easy to study, and will also show up impacts quite quickly. We sample the ecology using three different techniques. We sample predominantly the infauna with a Hammond grab, and that takes a scoop of the seafloor. And then we sample the epifauna with a beam trawl, which scrapes the top of the seafloor. And then we also take seabed images, and this puts the smaller Hammond grab samples into a wider context and also lets us see how those components of the marine community are arranged on the seafloor. Once the sediment sample has been collected with the grab is retrieved and brought onto the ship. Here we take the sediment sample out of the grab, and we take a photograph of the sample so that we can look back at the sample later on and see what the sediments were like and see if we can identify any obvious fauna. We then take a subsample of the sample, which we analyse for the sediment composition, and then we sieve the sample to remove any of the fine material, and the rest is then fixed with chemicals and taken back to the laboratory where it's analysed. Once the trawl sample arrives on the vessel, first we take a photograph of the trawl sample and we measure the volume of the whole catch, and then we sort the catch into different species and take photographs of them and identify them to species level where possible. With some of the smaller animals where it's quite difficult to identify them without a microscope, they're preserved and taken back to the lab for verification. So once the grabs come back to the lab, first of all the chemicals are washed off, and then the animals are separated from any remaining sediment using a microscope. And once all the animals have been picked out of the sample, they're separated into the 5 major phyla, so Annelida, Crustacea, Mollusca, Echinodermata, and anything else put into another pot. And then each of these pots is identified in turn, so that every organism in there is identified and counted. And then once they've been counted, they're also biomassed. And then this data all goes into a database here and is analysed later. A biotope is made up of 2 components, the biological community and the physical environment in which the biological community exists. These are classified using environmental parameters such as the sediments on the seafloor, the amount of energy in the systems, that might be waves or tides, and then also the amount of sunlight getting to the animals on the seabed. So in the intertidal zone, there's sunlight which reaches the seabed and you get green algae, and as you move deeper you get red and brown algae, and then those disappear where there's no sun reaching the seafloor. So that all of those different components make up a biotope. Within the REC projects, we use the UNIS habitat classification scheme in order to classify our biotopes. This scheme was developed by the European Environment Agency and is a hierarchical classification scheme. The system starts off splitting between marine habitats and terrestrial habitats, and then the marine habitats are split further using physical environmental factors such as depth, sediment type, and the amount of light reaching the seafloor. Because we're quite limited in the data we can collect in the marine environment, we've started modelling marine biotopes. Because biotope classification schemes are hierarchical, this allows us to take modelled environmental data as the base of our model. So we use oceanographic model, which model the routes of waves and tides, and we combine these with sediment maps, which have been created using sample data, but give a continuous map of the sediments on the seafloor. We combine these layers all together to give a description of the physical biotopes. And once we've described the physical biotopes, we can then take our point data, which gives us information on the biological communities, and we can see where these occur most regularly, and we can then extrapolate that data to produce a combined biotope map of the seafloor. The REC programme was designed to provide regional maps of the seafloor. This will include maps of the sediment type, the biological communities, so different animals, a distribution of rare species, and also archaeological features. And the way that these maps will be used in the future is to inform marine planning. So marine planning is a process by which government decides which areas are a priority for which activities. So that might be conservation, or it might be fishing, or it might be a wind farm. And all of these activities have conflicting needs, and by mapping the resources that are there, it allows us to plan these activities so that they have a minimal impact on the environment. Certain marine habitats have been identified as being particularly important for conservation, and this can be for a number of reasons. Usually it's because they're particularly rare. It may not be rare in the UK, but they may be rare in a European context. And the other primary reason is that they support a high biodiversity of other marine life. And these have been identified under the Habitats Directive, which is an EU directive, under Annex 1, which is a list of habitats which are particularly important for conservation. So the 3 most common Annex 1 habitats that we find offshore in the UK are sandbanks, rocky reefs and biogenic reefs, and these were found in the RECs. Examples of biogenic reef included the ross worm, Sabellaria spinulosa, and also mussel beds, Mytilus edulis. And rocky reefs is basically anything where there's rock protruding through the seabed, and these areas support particularly biodiverse communities, including sponges and bryozoans and hydroids. And then the other habitat is sandbanks, and these are particularly important because they provide a habitat for sand eels, which are a very important component of marine food webs. Alien species are animals which have been introduced from outside UK waters. These usually come in ballast water in ships, but they can also be introduced in aquaculture products. One example of this is Crepidula fornicata, the American slipper limpet, which was introduced first in 1872 in Liverpool Bay. This limpet came in with the American oyster, which was introduced on purpose to remedy a decline in the native oyster fishery. Limpets have been found to have quite a detrimental effect on oyster fisheries, and this is because they form very dense aggregations. And where they form dense aggregations, they cause more fine material to collect in between the individual limpets, and they also produce feces and pseudo-feces, which increase the fine sediments. And this makes the substrate unsuitable for settlement of our native oysters. During the Humber REC, we discovered a series of tunnel valley features, which are deep seabed depressions, which are thought to have formed during subglacial erosion, so erosion which occurred during the melting of the ice sheets. These features support quite unique communities, and we studied two of them in detail, which is the Silver Pit and the Sole Pit. What we found was extensive Ross worm reefs on the western flanks of the Silver Pit, which is the largest of the tunnel valleys. And these reefs are obviously an important conservation habitat, so this was a really important resource to find. In the Sole Pit, which is much more sandy, we found very high densities of bivalve communities. And the importance of these pit features is not only the habitats that exist within them, but also their physical structure. Because they're quite deep, they're very unlikely to be impacted by anthropogenic activities. A food chain is a group of animals which are related because each member of the food chain is eaten by the one above it in the food chain and eats the animal below it in the food chain. So it's just a single chain of animals which essentially eat each other. So a simple example, in the marine environment, most food chains start with the sun, and photosynthetic organisms take their energy from the sun. And in the marine environment, that's phytoplankton. Phytoplankton are eaten by zooplankton, which are small animals which live in the water column. And then the phytoplankton are then eaten, for example, by benthic organisms, which are then eaten by small fish, which are then eaten by big fish, which are then eaten by humans. And that's an example of a food chain. Several food chains make up a food web, and there can be interactions between food webs. So you might get lots of different big fish eating the same little fish, for example. Marine food webs sit within marine ecosystems, which also incorporate physical cycles such as nutrient cycling and water cycling, and also encompasses the habitat which supports the marine food web.